Well, wherever you are, good morning, good afternoon, good evening to MycoTalks. My name's Neil Gow, and I've honour to be linking up with Oliver Kurtzai today to chair the two talks we have today, which are from Julia Koehler from Harvard Medical School and Jim Kronstad from UBC, British Columbia. And we've got two terrific talks in prospect. So as you know, MycoTalks takes topics of medical mycology around the world. And the idea of the project was inspired some time ago, and it had its fantastic place in communication during the COVID pandemic. We always have two 30-minute talks, one after the other, followed by 30 minutes of discussion immediately after both talks. And you are very encouraged to submit questions that we will present as moderators to the two speakers. And please do that using the Q&A tab that you have there. They'll be seen by us, and we'll be able to relay those to the speakers after you've heard the two talks. The session today, like others, will be available on the MRC Center of Medical Mycology YouTube channel at Exeter University, and you'll be able to find these and other ones to that link. I also wanted to mention to you that there are going to be some additional MycoTalks for you to look out for. On October the 31st, as you see here, Gordon Brown and John Taylor will be speaking. And then on the 12th of December, we'll have David Moyes and Rob Kramer. So look out for those ones as well. Also, just wanted to mention to you that we have some other offerings which will be of interest to many of you. There's the trainee seminar series, which is for students, postdocs, and clinical trainees and new faculty. And you can present your work to a big audience around the world. These are on the second Thursday of the month, but you can also make some adjustments for this. So watch out for those as well. And also to remind you that we have the myco clinics with a more clinical emphasis. And there's one to look out for on the 3rd of October, which will be about the use of new antifungals in the clinic. So if you're a clinical medical mycologist, that's something for you. So now I'm going to stop sharing this and tell you a little bit about our first speaker, Julia Koehler. And Julia's title today is For Signaling Stress Management Phosphate Acquisition are Linked in Candida albicans. I've known Julia, had the pleasure of knowing Julia for some time. She studies Candida albicans predominantly, nutritional and stress signaling and their impact on the organism's pathogenesis and drug response. She started her career as an MD in Heidelberg, which is her home in Germany, and she was trained in pediatrics and pediatric infectious disease at Boston's Children's Hospital and stayed on to care for immunocompromised children with complex infections there. And at that time, she started to train in molecular genetics in Jerry Fink's lab at MIT in Whitehead, and she studied the regulation of hyphal growth. And she invented something many of us use, spider medium. And then she started her own laboratory at Boston's Children's Hospital. And in her clinical work, she works to understand more deeply how Candida cell program responds to various host environments and is needed, that's urgently needed as a basis to generate and design better therapeutics. And our current post, she's an associate professor of pediatrics at Harvard Medical School. So Julia, you're very welcome, and we look forward very much to hearing your talk. And I'll now hand over to you and look forward to your talk on TOR signaling and stress management. Thank you very much, Julia.
Thank you so much, Neil. It is a huge pleasure and a great honor to present at MycoTalks. I am just gonna share my screen. One sec here. Oops, sorry. Okay,
Perfect, Gillian.
yes, technology. So as Neil mentioned, I want to tell you about the connection between target of rapamycin and phosphate signaling that my lab has been working on. And first, I'd like to acknowledge the wonderful people who did this work and also my amazing collaborators. Tamina Chowdhury is a hugely gifted scientific thinker and inspired our ongoing tour work when she analyzed the connections between target of rapamycin and protein kinase A signaling in the lab. Ningning Liu is an extremely hardworking, tremendously talented former postdoc who now has his own lab in Shanghai. Catherine Chi is our brilliant Candida whisperer. She is a deeply thinking investigator who cracks every tough question that we ask the Candida cell. Michael Acosta Zaldivar is a former postdoc in the lab, a genius of hard work and of unlimited scientific curiosity. Nikita Jani and Udita Roy, both very talented scientists who each made contributions to this work. And I'd like to acknowledge our wonderful collaborators at Children's Hospital and far and wide everywhere in other institutions without whom this story would not have come together. Candida albicans is the most commonly isolated human fungal pathogen. It most commonly occurs as diploid cells, which grow as budding yeast or filamentous pseudohyphae or hyphae, and switches between these cell types are regulated by a number of signaling systems that I'm sure many of you are working on yourselves. And importantly, by the target of rapamycin complex 1 signaling system, which I am going to be calling TOR or TORC1 going forward. So TOR is a system that collects information about favorable versus unfavorable intracellular and extracellular conditions in all eukaryotic cells. It uses that information to make and to implement decisions about growth, favoring growth and proliferation versus about inducing survival and stress responses in unfavorable conditions. And the TOR1 kinase or the TOR kinase at the center of this system was discovered by our own Joe Heitman, who used rapamycin as a tool for his genetic analysis. That was when he was a postdoc in Michael Hall's lab, who called TOR the brain of the cell. I like that metaphor, and that's how I think about it. And so today I'll tell you how we took a maximally unbiased approach to discover novel roles of TOR, and that led us to investigate phosphate homeostasis. We wanted to use a minimally biased approach. So while at the time there were TN5 and TN7 transposition systems in Candida albicans, we decided to start over and use the Mariner transposon because it needs only the dinucleotide TA as a recognition signal for its cut and paste transposition system. And we marked a Mariner transposon with our dominant selectable marker NAT1, in vitro transposed this into a Candida genomic library, used that to generate about 14,000 heterozygous transposon insertion mutants, and found a rapamycin hypersensitive mutant to have an insertion in the open reading frame that encodes a homologue of the phosphate transporter well studied in Saccharomyces Fo84. When we generated homozygous deletion mutants, null mutants in Fo84, they recapitulated that rapamycin hypersensitive phenotype, and they also had a severe growth defect on medium where they were starved of phosphate. We used our readout of TORC1 activity, which is the phosphorylation state of ribosomal protein S6, to look whether Fo84 null mutants signal normally, and found that they don't. So at every distinct phosphate concentration made available to these cells, the cells lacking Fo84 had a diminished TORC1 signal. We wanted to understand how a signal or how information from Fo84 might actually arrive at TORC1. Candida albicans was known to have homologues of 2 GTPases that activate TORC1 in mammals versus in Saccharomyces respectively. So we looked at null mutants in these GTPases and found that lack of RHB1 did not affect the TORC1 response to phosphate availability, whereas lack of GTR1 did affect that response. So we concluded that GTR1 might transmit that signal to TORC1. If this is the case, a strain lacking Fo84 that overexpresses GTR1 or overexpresses a constitutively active GTR1 might suppress the growth defect of Fo84 null cells on rapamycin. This is in fact what we found. So the simple overexpression alone of GTR1 suppressed the poor growth of Fo84 null cells. When we then looked at the TOR signal, we found that actually Fo84 null cells that simply overexpress GTR1 recover their phospho-S6 signal intensity, suggesting that the phosphate signal from Fo84 might be routed to TORC1 through GTR1. Does a reciprocal regulatory system exist from TOR1 to Fo84? To look at this, we use 2 sets of mutants in the essential TOR1 kinase. Both of them have deletions of 1 TOR1 allele. One of them has the otherwise wild-type TOR1 open reading frame under the control of repressible TETO, such that when we add a drug, this transcription is shut off. The other set of mutants contains a truncated TOR1 open reading frame such that the N-terminal 381 amino acids are not expressed in the protein product. And these amino acids represent a protein-protein interaction domain called heat repeats that is the least conserved region of the Candida TOR1 with the human TOR kinase, and that's why we were interested in this region. And when we looked at FO84 expression in cells that lack these heat repeats, we saw that unlike the wild type, which when cells are transferred into rich medium with lots of phosphate, represses FO84 expression, cells lacking these N-terminal heat repeats fail to repress FO84, and even TOR1 full length is required for that repression because the cells fail to repress transcription when we deplete TOR1 from TET-O. So we thought that we can model these relationships with this in this way that I'm depicting here. A regulatory role of the big system phoregulon toward FO84 was previously known from Saccharomyces, and my blue arrows show the regulatory relationships that we discovered, namely a signal, an apparent signal, I'm going to say, from FO84 to GTR1 that was known to activate TORC1, and a signal from TORC1 back to FO84 that very likely goes through the phoregulon. Now, TORC1 co-regulates stress responses, as I mentioned, and these are particularly relevant for Candida's host interaction. So we asked whether FO84 plays a role in some key stress responses. We found that, in fact, mutants in FO84 are hypersensitive to both cell wall and oxidative stress. And what I'm showing you here are cell wall disruptive conditions, for example, exposure to Congo red that binds to and disrupts chitin fibrils. And we saw that the FO84 null mutant is hypersensitive to these cell wall disruptive conditions. When Louise Walker and Neil Gauss group examined these mutant cell wall architecture by transmission electron microscopy, she found that the FO84 null mutant cell wall outer layer is significantly thinner than that of wild type. And when we then looked at specific components of the cell wall that lend it structural stability, like chitin and beta-1,6 glucan, our quantitation showed a diminished chitin content, a diminished beta-1,6 glucan content, and also decreased phosphomannan in FO84 cell walls that led us to believe that these are simply thinned and weakened cell walls. Now, turning to oxidative stress, superoxide-inducing compounds like plumbagon and menadione strongly affected FO84 null cells and caused a substantial growth defect, as did peroxide. So yes, FO84 cells are hypersensitive to oxidative stress as well, and that affects their interaction with, our liquid tissue, which is blood. So whole blood, even whole blood, kills FO84 null cells at a much greater percentage than wild type or reintegrant cells, and isolated human neutrophils, which use oxidative stress to kill the Candida cell, do the same. Per Leungdahl and Ilva Engstrom's lab used a Drosophila model to test virulence of these cells given our oxidative stress phenotypes, and in fact found attenuation of virulence. And Scott Filler's lab used a oropharyngeal candidiasis model to quantitate the fungal burden in tongues of infected mice, which was significantly decreased for the Fo84 null cells. Jose Lopez Rebo used these cells and found that in a systemic candidiasis model, they are also attenuated, not, abrogated, but attenuated in virulence. So thinking about these defects in two significant, host-significant stress systems, we were wondering, phosphate, what is up with that? Do these defects share the same mechanism? Is it just lack of phosphorus for the cells? Is it the disturbed TORC1 signaling that is causing these defects? So when we looked at oxidative stress, we saw that for important phenotypes, Fo84's null mutants oxidative stress defect is independent of the phosphate that they have available to them. Here we're looking at reactive oxygen species inside the cell, and the Fo84 null mutants still have significantly increased reactive oxygen species in excess phosphate medium as they do during phosphate starvation. However, an important superoxide dismutase of the Candida cell is SOD3. It dismutates superoxide, damaging reactive oxygen species into less damaging peroxide and water. And we found that the protein level of this SOD is decreased in Fo84 null cells. That seems to be a regulatory defect because nutritional signals that normally induce or repress SOD3 in wild-type Candida had the same effect in the null mutants. Here we're using an antibody generously shared by Val Culota to measure SOD3 protein levels. This regulatory defect seems to be related to TORC activity because when we overexpressed GTR1 in Fo84 null cells, they recovered their SOD3 protein level as opposed to vector-only expressing cells. So we model that Fo84 has this activating effect on GTR1 that we hypothesized before. Therefore, TORC1 is activated and regulates SOD3 expression, which is then available to block the harmful effects of superoxide released on the Candida cell by host neutrophils. Turning now again to cell wall stress, contrary to our oxidative stress findings, we found that the effects on the Candida cell do depend on the phosphate that they have available to them. In moderate phosphate concentrations, Fo84 null cells have a significant growth defect, but in excess phosphate, they grow almost as well as the wild type. And that is true even for double mutants with cell wall biosynthetic enzymes like chitin synthase 1 that we created in the Fo84 null background. Even they recover growth when we feed the cells excess phosphate. So the cell wall defect seems to depend on the phosphate available to the cells. We looked at TORC1 activity. Does that help them withstand cell wall stress? And we found that Congo red still has a very strong growth limiting effect on FO84 null cells, even when they overexpress GTR1, suggesting that TORC1 activity does not contribute to FO84 null cells' lack of ability to withstand cell wall stress. We used metabolomics to get at the mechanism of the cell wall stress defect. And here are metabolites that are building blocks for cell wall biosynthetic enzymes, UDP glucose for beta-1,3 and beta-1,6 glucan synthase, UDP and acetylglucosamine for chitin synthase. And when metabolomic mass spec quantitated a number of these intermediates, what we found that before significant phosphorylation steps, there was a backup, there was an increase of metabolites in FO84 null cells compared to wild type cells, whereas after phosphorylation steps, FO84 null cells had decreased intermediate metabolites and decreased sugar nucleotides that are required for cell wall biosynthesis. And independently quantitating the ATP concentration in these cells, we found that it was decreased, suggesting to us that it is a simple lack of phosphate that blocks these cells' ability to produce the substrates that they need to build strong and resilient cell walls. So our model here is that FO84 permits sufficient phosphate acquisition for intermediate metabolism to produce the building blocks for these cell wall biosynthetic enzymes. And when cells lack these building blocks, the resultant cell walls are going to be thinner and weaker. We then wanted to ask the cells another question, because phosphate is such a crucial element for construction of macromolecules that all cells need. DNA, RNA, ribosomes, and therefore proteins all require phosphate. So we used another open-ended approach, which was simply to take quadruple mutants in all the inorganic phosphate transporters that Candida has, constructed by Catherine Chi, and passaging these quadruple mutants that are very defective in phosphate uptake over 30 passages in moderate phosphate concentrations. There are two separate lineages of two independent transformants. We saw that one lineage reacquired an almost wild type ability to grow in these phosphate limiting conditions. The other lineage also recovered fitness to a significant extent, but never got back to wild type level fitness. Abhishek Mishra in Matt Anderson's lab sequenced the genomes of our wild type progenitor, then our quadruple mutant ancestors of these evolving lineages, as well as selected populations from early versus late passages of these two quadruple mutant lineages. What we found was that in chromosome 2, both lineages independently acquired trisomies, and in both lineages, those trisomies started to resolve back to diploidy during evolution and during acquisition of more point mutations. In lineage 2, a residual segmental trisomy remained at the end of the experiment. And when we looked which open reading frames reside on this remaining trisomic segment, we found that they include TOR1 and a TORC1 associated kinase RCK2. So in summary, we started with an open-ended question: what are some novel activities in the cell associated with TORC1? And the cell told us phosphate homeostasis. We then asked the cell, can it adapt to phosphate scarcity in another open-ended approach where the cells will tell us what they want? And that question led us back to TOR1. So in summary, PHO84 appears to have an activating activity towards TORC1, which then allows the cells to successfully deal with reactive oxygen species. PHO84 also provides metabolism of the cell with the phosphate needed to construct robust cell walls. And in reality, these two roles of PHO84 are not separate processes. Of course, they are going to have interactions at multiple levels, and it is, our interest and hopefully the fields to discover in new and exciting work where these interactions occur and how they happen. And with that, I want to end and hand it back over to our organizers, whom I'd like to thank again for the opportunity to speak with you.
Yeah, thank you very much, Julia, for these insights into a complex signaling network. And as always, we will have a chance to discuss your talk after both talks. So you get a minute rest, and anyone can please submit questions through the Q&A function, and make sure to just indicate whether your question is for Julia's talk or for the talk that is now going to come. And for this, it's my great pleasure to introduce our second speaker. And this is Professor James Kronstad. He is a professor in the Michael Smith Laboratories and at the Department of Microbiology and Immunology in Vancouver at the University of British Columbia. He has been there for quite some time, but before that, he completed his postdoctoral positions at a company, Zymogenetics, and also at the University of Wisconsin in Madison. And he is interested in understanding mechanisms of fungal pathogenesis, and I think that the exclusive characteristic of his group is that he focuses both on plant pathogenic fungi, specifically Ustulago-Mides, and human pathogenic fungi, Cryptococcus. And I think this is really something that we have probably too rare in the field. Today, he's going to talk about Cryptococcus neoformans and the role of glutathione-mediated redox regulation in virulence. Jim, it's great to have you here, and the stage is yours.
Okay, thank you, Oliver, and thanks very much to the organizers for the opportunity to speak. I very much appreciate this. So what I'm going to do today is focus on a couple of recent studies that have to do with connections between mitochondrial signaling and virulence factor elaboration in Cryptococcus neoformans. And the work that I'm going to talk about, as Oliver mentioned, was performed in the Michael Smith laboratories at UBC in Vancouver. So also, as Oliver mentioned, we work on two different systems for studying pathogenesis. And our central question has to do with how fungal pathogens sense nutrients to proliferate in host tissue and also to cause disease through the elaboration of virulence factors, again, addressing pathogenesis in plant and animal hosts. So I won't say anything about Eustallago-Matus other than to point out that it's a biotroph where interesting features of biotrophy are nutrient dependent. And we recently discovered that mixed carbon sources regulate biotrophic phenotypes in this pathogen. But today I'll focus on Cryptococcus neoformans and really think about aspects of melanin formation and capsule formation as they are influenced by mitochondrial function and by nutrients such as iron and iron limitation. Okay, so many of you already know, and I'm sure you've had many talks before about Cryptococcus and cryptococcosis. I'll just remind you that cryptococcal meningitis is a very serious problem in people who are immunocompromised, as highlighted by the World Health Organization's critical pathogen priority group, of which C. neoformans is in the top 4. And we all acquire these fungal infections by inhalation of fungal cells. And in immunocompromised people, the cryptococcus can disseminate from the lungs to the brain to cause this life-threatening meningitis. These key virulence factors include melanin formation on catecholamine substrates, capsule formation that I mentioned in response to host conditions, and then also the ability of the fungus to survive at 37 degrees and to proliferate in and hide out in macrophages. And of course, macrophages are thought to contribute to a Trojan horse mechanism by which the fungus can disseminate to the brain. So we spent many years characterizing iron transport functions and iron regulatory functions to begin to understand that iron regulation of capsule formation. And I won't go through all of these different iron uptake systems and all the regulatory factors, just to point out that host sources of iron, such as transferrin, can be used by reductive iron uptake systems. Cryptococcus has siderophore transporters to steal siderophores from other microbes. It does not make its own siderophores. And it has an uptake system to use heme, the most abundant iron source in mammalian cells, mammalian hosts. And we don't know everything about this system, but we're actively trying to characterize it. So today, what I'll focus on first is aspects of glutathione-mediated redox regulation. And glutathione has an important signal that comes from mitochondria along with iron sulfur clusters to influence regulatory factors that control the expression of these iron uptake functions, as well as a number of other metabolic functions important for virulence. And the transcription factors CIR1 and HAPX will play a prominent role in the second part of the talk. And this glutathione-redoxin, this monothioglutaredoxin GRX4 interacts with these transcription factors and plays an important role in regulating the iron uptake systems and a number of other factors. So I'll focus on glutathione first and then come back to these regulatory factors in the second part of the talk. So what we've been thinking about in this study has to do with glutathione signaling from mitochondria. And we know that glutathione is exported along with iron sulfur clusters from this ATM1 protein, which is an important protein for virulence for Cryptococcus neoformans. And it influences the iron regulatory network through GRX4 and CIR1. So one of the key outcomes of this regulation is the expression of the lacase genes that encode the genes that encode lacase for melanin formation. And melanin, of course, is important for the fungus in the environment and also in the mammalian host. So we focused on glutathione because of this connection with the mitochondria. And it was also of interest because glutathione is a signal in bacterial pathogens, Listeria, for example, to influence virulence gene expression. And there's elegant studies from Dan Portnoy's lab at Berkeley that illustrate this for Listeria. So this kind of evidence for glutathione prompted us to really looking in detail at this molecule as a key redox regulator and redox homeostasis molecule in the cell. So in collaboration with Arturo Casadoval's lab, we set out to make mutants in the glutathione biosynthesis pathway to really address this question of whether glutathione is important for virulence, for cryptococcal disease. And we knocked out GSH1, the first gene in the pathway, the two-step pathway for glutathione synthesis. These mutants are quite sick and difficult to work with. So we moved on to the second gene in the pathway, GSH2, which includes glutathione synthetase, combining gamma-glutamylcysteine and glycine to make glutathione. And the important phenotype that came out initially that we really focused on was that two independent mutants lacking this enzyme don't make melanin on a substrate L-DOPA. So this melanin defect automatically gave us relevance to virulence for Cryptococcus neoformans and prompted us to dig more deeply into what glutathione does to contribute. So the first thing we did was confirm that the mutants, two independent mutants, don't make glutathione when the gene is missing. So compared to the complement and a wild type strain, there's very little glutathione present. And we also characterized the ability of the mutants to grow on various media. You can see that on yeast nitrogen base, minimal medium, the GSH2 mutants are oxytrophic. They don't grow very well. And they're oxytrophic for glutathione or for the oxidized form of glutathione, GSSG. And they grow fine on rich medium YPD. You can't complement that or rescue that oxytrophy with methionine or cysteine. You can rescue it a little bit with ascorbic acid, but heme and iron do not rescue growth on OIMB. So that was a little bit of a puzzle. We still don't fully understand the contributions to iron homeostasis at the level of growth, but as I talked about later, we have connections with the iron regulatory network. One thing we tested early on as well was whether or not Cryptococcus can export glutathione into the medium. And we did this by using a spent medium experiment where we grew wild type or mutant cells and then used the supernatant from cultures to test whether the mutants could grow. And wild type or complement strains, the supernatant, the spent medium allows the mutants to grow. Whereas if you use the spent medium from the mutants themselves, the mutants don't grow as you might expect. So that along with the glutathione depletion, this is evidence that glutathione is exported from cryptococcal cells and could have signaling roles that need to be investigated. We also looked at virulence as predicted by that melanin defect. We thought there would be a virulence defect in mice. And so we did the survival analysis, inoculating mice by intranasal or by inhalation using the inhalation model. And you can see that the GSH2 mutant is attenuated for virulence compared to the wild type and the complement, which is only partially complementing. And what's interesting, when we looked at the fungal burden in these mice at the time that they succumbed to infection, we found there was very low levels of the mutant in the brain, kidney, and blood of the infected mice. And so it looks like there might be a problem. At that stage, it looked like there might be a problem with dissemination for the GSH2 mutant. We did a time course from 7, 14, 21, and 26 days of infection to look at that possibility that there's a defect in dissemination. And over time, you can see as comparing the GSH2 mutant here in light blue with the wild type in pink or red, that the mutant really is not able to persist in the mice at these later time points compared to the earlier time points, and particularly disappears from the brain pretty early on during infection. So there may be an impaired dissemination as well as the the attenuated virulence for a GSH2 mutant. One interesting aspect of the infection that we noted when we looked closely at the histopathology is that the GSH2 mutants appeared to not make these titin cells that have been observed in pulmonary infections for Cryptococcus. And so this is a unique morphological phenotype where the cells become quite enlarged. Some population of the cells become enlarged during the pulmonary infection. And you compare that with the complemented strain or the wild type, where you can find these enlarged cells, these so-called titin cells, in an infected tissue. And we recovered the cells and looked at their sizes and quantitated the sizes to get an estimate of how different they were. And you can see that the mutants are quite a bit smaller than the titin cells in vivo, and we can reproduce this in vitro using conditions that several groups have described for generating titin cells in culture. So again, the GSH2 mutants are defective in making titin cells. Now, we have a series of experiments where we began to look at the defect in melanin formation as a key feature of the mutant. And so we've done a number of experiments to really dig into why there's a melanin defect and what the underlying mechanism might be. So we quantitated melanin both extracellularly and cell wall bound melanin, and could see the defect in the mutants compared to wild type and complement, recapitulating quantitatively what we see in this L-DOPA plate assay that I showed you before. And we also checked other virulence factors. There's no capsule defect in these mutants. There's not really a temperature sensitivity, but they did have a defect in urease production, which is interesting because urease, that enzyme, is thought to be important for facilitating dissemination of Cryptococcus to the brain. So a contributor to the virulence profile for the pathogen. And we noted, and again, when we were collaborating with Arturo's group for this study, we noted that his publication recently described this reciprocal modulation of ammonia and melanin production for Cryptococcus neoformans, suggesting that there is a pH effect, perhaps, that might contribute to this defect we see with the GSH2 mutants. So we tested that idea by looking just simply at the pH of the culture supernatants from wild type and the mutant. And of course, we found that the mutant is quite acidic compared to the wild type in the culture supernatant. So one contributing factor to this melanin defect may be this pH difference, extracellular pH difference at the site where melanin is formed in the cell wall. So we tested that by growing the cells in the presence of a buffer, MOPS, and you can see just in a simple visual look at culture tubes that you can restore melanin formation in the mutant by buffering the medium. And we could quantitate that for extracellular melanin and cell wall melanin to show that MOPS buffering allowed recovery of melanin production. So pH is one factor that appears to be contributing to this melanin defect. We looked more closely also at what was accumulating in the supernatant that might begin to understand that acidification and other aspects of the redox conditions that would support or impair melanin formation. So we looked just at total thiol content in the supernatant and found that the mutants have elevated thiol content, giving us a first clue that there might be major metabolic shifts due to the loss of GSH2 in these mutant backgrounds. And we also looked at what that impact might be of thiol accumulation in the supernatant, redox changes due to loss of GSH in a variety of different ways. So we looked at reduced reactive oxygen accumulation in the mutants. And you can see with treatment with hydrogen peroxide, the mutants do not accumulate ROS, this kind of assay, quantitative assay. But we do see enhanced antioxidant activity looking at superoxide dismutase activity, catalase activity, enzymes you heard about from Julia a moment ago. So the GSH2 mutants seem to have hyperactivity for antioxidant functions. Again, pointing to this idea that there's a major shift upon loss of glutathione in both intracellular and extracellular redox environments. The extracellular environment we could show has a very different reducing potential using this ABTS radical scavenging assay. And here you see that the two mutants don't decolor, they are able to decolorize, suggesting that there's elevated reducing activity, consistent perhaps with that accumulation of thiols. So then we dug even deeper into what's changing in the cell intracellularly and extracellularly for the mutants compared to wild type by doing some LC-MS experiments to really get at the metabolome and understand the differences in the metabolome between the two different strains. So just in a broad scale look at what pathways change between mutant and wild type and what we see in the metabolic profiles. So we see a lot of changes in amino acid metabolism and secondary metabolites shown here, and then a variety of other functions, including carbohydrate metabolism and changes in other metabolites down below here. We focused in particular on the supernatant and looked at what was accumulating in the mutant. And here we can see that a number of different compounds, in particular a number of different amino acids, begin to accumulate in the supernatant upon loss of this GSH2 activity. And included in this supernatant analysis, we found a lot of organic acids, dicarboxylic acids that might explain that acidic pH change that we saw before. In contrast, the wild type seems to accumulate amino acids in the cell extracts to a higher level than you see in the mutant. The wild type also appears to accumulate other molecules in the supernatant, including ergothioneine, which had not been seen before and could be a substitute for glutathione. Now, interestingly, when you really dig into the metabolites that are different between the wild type and the mutant in the supernatant, you can find a number of compounds that are known from the literature to be antioxidants. So these glucuronides shown here are much higher in the mutant than in the wild type. Other molecules like caffeic acid, which have antioxidants properties, are higher in the mutant than they are in the wild type. And then you can find molecules that are higher in the wild type, like ergothione that I mentioned, or reduced glutathione as shown here. So we have this idea that there's a major shift in the metabolism and metabolites that accumulate in the supernatant when you lose glutathione intracellularly. So this brings us to this idea that in a wild type cell, glutathione's contributing to this redox balance, a neutral pH at the exterior surface, and that allows the fungus to melanize. Lactase activity and spontaneous chemical reactions that result in lactase or in melanin deposition in the cell wall. In contrast, loss of glutathione due to this mutation, then it leads to accumulation of thiols, antioxidants, and acids that result in this acidic pH in the supernatant, and that impairs melanin formation. And we tested just whether extracellular GSH would have an influence on melanin formation in wild type versus the mutant. And you see that there's a Goldilocks zone for melanin formation in response to glutathione, where there's this region where concentrations support it and higher levels do not. And you can recover the GSH mutant phenotype for melanin formation also, but not exactly to the same level. All right, so that's what I wanted to say about glutathione in the context of melanin formation. I want to come back to this communication between mitochondria and the iron regulatory network. And we know we can use melanin as a readout and lac gene expression as a readout to begin to understand regulation of melanin formation, but we really want to know what are the signals and how does the cell interpret nutritional signals to control this network or to influence this network. So I'll focus on CIR1, this transcription factor, and the other iron regulator HAPX. And I'll just show you up front that a long time ago, we discovered that CIR1 regulates all the known major virulence factors of Cryptococcus neoformans, including the capsule, shown here, temperature, shown here, and melanin formation. And I'll just note that CIR1 is a repressor of melanin formation, and there's a glucose response there. And of course, given those phenotypes, a CIR1 mutant is lacking for virulence in mice. The monothiol glutaredoxin GRX4 also is required for virulence and is a key interactor, as I'll show you, with this transcription factor CIR1. Okay, so we went on to do with CIR1 a couple of years ago in collaboration with Won Hee-young and Unsu Do in Korea, to look at the direct targets of CIR1 and the other iron regulator HAPX. So we used ChIP-seq to identify the direct targets for these proteins, and then used RNA-seq to identify the transcriptional changes associated with those direct targets. And I won't go into all of the details of all the different functions that are regulated, but there's a wide variety of functions for CIR and for HAPX. So I'll just note that CIR1 represses LAC1 for lacase. So that's a direct target of regulation and a useful reporter for CIR1 activity. HAPX, in contrast, is a repressor of iron-requiring functions and mitochondrial functions, and also for oxidative stress proteins, the response to oxidative stress, and also regulates a few iron uptake functions, including siderophore transporters. So we have this platform now of understanding what's regulated by these transcription factors, this iron regulatory network. The glutaredoxin GRX4 also influences similar kinds of functions related to what we see with HAPX. So there's a close resemblance of what HAPX regulates, including things related to oxidative stress and mitochondrial function, shown here, and iron sulfur cluster function, heme binding, those kinds of molecules, as well as a wide variety of other functions that are more related to the things that CIR1 controls. All right, so just to show you that GRX4 actually does regulate these things in a phenotypic fashion, comparing the electron transport carrier functions, we could show that a GRX4 mutant is sensitive to inhibitors of electron transport, including rotenone for complex 1, antimyosin for complex 3, alternative oxidase, for example. And it's also sensitive to these oxidative stresses, including the kinds of stresses that Julia just described, plumbagon, H2O2. So all these phenotypes lead us to think about mitochondrial functions and their connections with the iron regulatory network. I should just point out briefly that GRX4 interacts with CIR1 and HAPX in a yeast-2 hybrid assay. So we could demonstrate that, and we confirmed it by affinity purification and mass spec. I won't go through all of that, just point out that the GRX motif with the cysteine residue is important for that interaction. What's more interesting, I think, is to look at GRX4 as a reporter of signals that could come from mitochondria, and also signals that come from nutrients like iron and heme. So in this case, we have a GRX4 M-cherry fusion that's localized to the nucleus in low iron medium. CIR1 GFP is also in the nucleus in that condition. But if you add heme or iron chloride, an inorganic iron source, then GRX4 partially relocalizes to the cytoplasm. So there's this localization response that happens in response to iron. And when we quantitate this, we can show that that relocalization happens in response to antimyosin, a complex 3 electron transport chain inhibitor. It happens in response to oxidative stress from H2O2. Happens also with a defect in the ATM1 mutants that ATM1, to remind you, encodes the transporter for iron sulfur clusters and glutathione from the mitochondria. And then when we do the yeast-2 hybrid experiment, we can show that hydrogen peroxide interferes with the interaction of GRX4 and CIR1. And that makes sense because you have that T cysteine residue in the GRX motif of GRX4 CIR1 transcription factor has two different cysteine-rich regions, which may be in thiol-responsive elements for responding to redox conditions. HapX as well has cysteine-rich regions. So we went on then to look at this phenomenon in more detail about what's the connection between electron transport and the activities of this iron regulatory network generated by GRX4 and CIR1 or GX4 and HapX. So in this experiment, which we published recently, we looked at complex inhibitors for the electron transport chain shown here. And we could find that these inhibitors, particularly known for complex 1 and antimyosin and mixothiazole for complex 3, interfere with melanin formation. Kind of going back to that idea that there's a redox balance necessary for the cells to make melanin and deposit it in the cell wall. What's interesting is that if you do this same experiment with the CIR1 mutant or the HapX mutant, then you can find a recovery of melanin for the complex 3 inhibition situation, and a little bit for CIR1 for complex 1, although there's a growth inhibition shown there. So we dug into the mechanisms of this, and I won't go into all the details, but we think that there are two different things going on here. For HapX, we think that it's regulating the components of the electron transport chain. It's also regulating iron-sulfur cluster formation, and it regulates oxidative stress functions, so that in the absence of HapX, these functions are elevated in expression, and we've shown that, and that might allow recovery of melanin by kind of reestablishing redox balance. And then for CIR1, I told you it's a negative regulator of the genes for lacase expression, shown here by quantitative PCR. So in that case, it may be de-repression of lacase that's partially restoring melanin formation. Okay, so we have this model where we have mitochondria responding to iron, generating these signals, iron-sulfur clusters, glutathione, leading to this influence on these iron-responsive transcription factors. So we end up with this model, this kind of complex network where mitochondrial functions are responding to various external signals. We know L-DOPA, the substrate for melanin, influences mitochondrial gene expression or the expression of genes for mitochondrial functions. You can inhibit these functions with antimyosin A and other ETC inhibitors. That influences redox homeostasis, which impacts melanin formation. And these transcription factors then are involved in regulating oxidative stress, and they're important for responding to these mitochondrial signals, including ROS and iron-sulfur clusters and glutathione. I should say that there are many other transcription factors and kinases that influence melanin formation, important ones discovered by Young-Sun Bond's group and some nice work published recently. Okay, so one last thing then, we have all these connections between mitochondria and iron regulation for iron uptake functions and these iron regulatory proteins. But from our ChIP-seq analysis and RNA-seq analysis with CIR1, we found that it also directly regulates functions for nitrogen acquisition, including amino acid permeases shown here. And these are, we know these are required for virulence based on work from other groups. Other aspects of nitrogen uptake are also regulated directly by CIR1. So right now what we're trying to do is build this model where we're thinking about mitochondrial sensing of host conditions, influencing the iron regulatory network and nuclear transcription, and then trying to understand how that influences mitochondrial function to support proliferation in the host. And of course, we're interested in looking at glutathione in more detail and reactive oxygen species in more detail, depleting glutathione in the mitochondria, and looking at those cis residues on these factors. Trying to integrate iron and nitrogen regulation, looking at CIR1 and GAT1 regulation, GAT1 being the nitrogen regulator, and then aspects of mitochondrial function. And of course, this is important for growth in the host because Cryptococcus is a strict aerobe. All right, just to summarize, I won't go through all of this in detail. We have characterized this monothioglutaredoxin and GRX interactions with the iron regulatory network and these transcription factors. We've characterized glutathione as a key molecule for redox homeostasis and a component of mitochondrial communication. Loss of GSH2 and glutathione causes a lot of different phenotypes, particularly impairs virulence and melanin and titin cell formation. There's a very dramatic remodeling of the extracellular metabolome upon loss of glutathione that requires further identification, characterization. Then we have these connections between the iron regulators and mitochondrial signaling. And we really want to understand more about that signaling, including how it relates to nitrogen metabolism and amino acid metabolism. Okay, so I'll stop there and I'd be happy to answer questions. I just want to acknowledge Braden Black, who did the bulk of the work on glutathione in collaboration with Arturo's lab, and really managed a great team of other investigators in our lab to help bring that story together. And then Pong and Eddie took the lead on studying connections between electron transport and the iron regulatory network, again, with a group of great people in the lab. And we have a very nice collaborative group that all works together very well. And just acknowledge these funding sources to support the Cryptococcus and Eustelago work. So thanks for your attention.
Thank you very much, Jim, and thank you very much, Julia. It's great to have a couple of talks about metabolic homeostasis and the ways in which fungus links their internal sensors to changes in the environment, because they are under attack. And I'm sure we will be able to explore that in a bit more detail. So I'm going to start with a question to question to Julia, and I'm going to take one of my own questions to start with, if I may, Julia. I know that you showed a very nice set of experiments where you took your cells and you exposed them to deficiency in phosphate and looked to see how they coped with that. And I wondered if you'd ever done any experiments with the exact opposite stress or signal, because certainly in certain media, which I think we've got lots of phosphate in it, you see that in the vacuole, you get these polyphosphate granules, and in the microscope, you can often see them dancing around by Brownian motion. And presumably these phosphate granules are, in metabolic terms, massive stores. And I wondered how long a cell would be okay before it ran out of these stores under conditions where they were either replete for phosphate or have an excess of phosphate, and whether you'd ever investigated that aspect, the corollary of the experiment you showed.
Yeah, that's a great question, Neil. Yeah, thank you for that. So fungi, including Saccharomyces and Candida, store excess phosphate that they have taken up in the vacuole. And that's because for them, phosphate is often limiting. I can go into why that is because I think it's pretty fascinating, but the phosphate stores in the vacuole that just consist of long chains of phosphates linked together are used, during phosphate starvation conditions, used up and extruded. There's a whole machinery for that. When we give cells excess phosphate in their medium, we don't observe stress phenotypes, and that is perhaps for several reasons. One is that they have lots of regulatory systems in their so-called phoregulon to block uptake of excess phosphate, including downregulating PHO-84, which is both the transcription, but also the localization to the plasma membrane of Fo84 is under tight control. So if Fo84 is not in the membrane, it's not gonna, when the cell has excess phosphate, Fo84 gets endocytosed, routed to the vacuole, and digested there. That process, we have watched that with GFP tagged Fo84, and it's very striking. That was Michael's work. So I actually think the cell has numerous redundant mechanisms to prevent both excess phosphate or orthophosphate in the cytoplasm, which would not be good because it's a strongly negatively charged anion, and also excess stores in the vacuole that would, let's say, overload the system, crowd out other molecules that need to sit in the vacuole. This Fo-regulon prevents that from happening. The 3 other phosphate transporters that Candida has in addition to Fo84 are less active, but we know from Saccharomyces that their homologs are also regulated by phosphate content of the medium. So long story short, I'm going to say the cell knows how to deal with excess phosphate in its surroundings. In real life, that's going to happen rarely, which I will be delighted to expound upon in response to perhaps another question. But in the lab, the cell will deal with it.
Great, thank you very much. I'll pass over to Oliver.
Right, and I continue with the first question here from the Q&A. Did you explore how the glutathione depletion affects dynamics and morphology of mitochondria?
That's a great question. We have not examined the influences on morphology or looking at fission or fusion, those kinds of dynamics that mitochondria might display. Mitophagy is interesting. We've just now started working on mitophagy, and we haven't looked at the glutathione mutants yet, but we do have a collection of autophagy mutants that do influence mitophagy. And the common trigger for us for mitophagy seems to work the best is nitrogen starvation. So it kind of takes us back to amino acids and thinking about what might be the important metabolic changes that could influence mitochondrial dynamics. So that's definitely an area we want to explore.
Great. And another question, I mean, how does the whole system behave once Cryptococcus is inside the host? Because I guess there's not much glutathione probably freely floating in blood or plasma, but some host cells might secrete it. I think the liver does secrete quite heavy amounts of it. So does this impact niche adaptation of Cryptococcus?
It could, yeah. We haven't really explored whether the fungus can be rescued by host glutathione, except to say that virulence is only attenuated. So it may be that there is enough glutathione around in host tissue to allow the cells to grow eventually and cause disease. So I wouldn't be surprised if that's what's happening. We weren't able to document any kind of phenotypic change as the readout of melanin when we looked at host tissue. That was pretty difficult to find melanized cells. We didn't really see that or weren't able to evaluate that. But yeah, I think that's an important consideration, is not only what's the fungus responding to, but if it's elaborating all these thiols and changing the extracellular pH, how does that influence its interaction in the host? And when we look at phagocytosis with bone marrow-derived macrophages, the glutathione mutants actually grow better in the phagocytes. So they might be conditioning that phagolysis environment somehow. I think that needs to be explored.
Okay, but did I get you correctly that the mutant, in addition to have the reduced virulence, has a specific defect in going to the CNS. Did I get that wrong?
Yeah, that's right. Yeah, it does seem to have a difficulty in dissemination in getting to the CNS and also other organisms too. It's other organs as well. It's not so good in blood. So it's the brain. We always focus on the brain because of Cryptococc's propensity to go there, but yeah, there are other organs affected too.
Great. Neil?
Julia, a question as to whether there's any role for TORC2 in phosphate regulation.
Yeah, thank you for that great question. So the TOR kinase in Candida is unique. There's just one TOR kinase, just like in humans, and it forms the core of two complexes. One is TORC1 that I was talking about. The other is TORC2. TORC1 is the complex. So, by the way, in Saccharomyces, there are two TOR kinases, just like Saccharomyces makes redundant paralogs, let's say, after the whole genome duplication. So these two complexes, TORC1 and TORC2, have somewhat overlapping and also distinct roles. TORC1, as I said, responds to nutrients and stress to regulate growth. That's the key thing it does, versus stress responses, shutting down growth and inducing stress responses for survival. TORC2 likely also responds to nutrients, but its role is to promote membrane biosynthesis and to regulate the actin cytoskeleton. So those two roles also are responsive to nutrients and regulate another aspect of growth, not just simply addition of these macromolecules, but where do those macromolecules go during the growth process. TORC1 is easier to study because it responds to rapamycin. TORC2, we don't have a specific targeting compound. So the two complexes are distinguished by specific components. TORC1 contains a large scaffold called COG1. TORC2 contains three proteins called AVO123. We have looked at COG1, but not at the AVO proteins or genes, because that would really be how we analyze the role of TORC2 in phosphate regulation. Interestingly to us, COG1 phenotypes recapitulate a lot of the TOR1 N-terminal truncation mutants, the one that lack specific heat repeats, suggesting that a lot of our observed mutant phenotypes are related to TORC1 and perhaps less to TORC2. So I think a rigorous answer to Faiza's great question is that we will need to mutate the specific TORC2 elements, the AVO genes/proteins, to see whether they have defects in phosphate handling. It's possible because without phosphorus, you can't build a membrane, or it's much harder to build a membrane. But I don't have the exact answer for you. That still awaits analysis of the very specific TORC2 components.
Thank you. That's very helpful. Okay, back to Oliver.
Right, Jim, there is a question in the Q&A from someone who knows more about mitochondria than me. This is about complex 5 and ATP synthase and how a membrane coupling is associated with the metabolic shifts you observed.
Right, that it sounds like they know a lot more about mitochondria than I do too. We have not looked at the ATPase and ATP and complex 5 or other aspects of membrane coupling. The approach that we've taken is to look at the endomembrane trafficking systems that connect mitochondria with the endoplasmic reticulum or with the vacuole. So the VCAP clamp complex and the VAM6 protein, looking at the impact of that protein on things like mitophagy. We're beginning to look at that, and also the ER mitochondrial encounter structures, looking at the components of that. So that's the approach that we're focused on right now in terms of mitochondrial communications with other organelles and trying to dig into that. But it's a good idea to look at brain coupling, and we do want the proteome of mitochondria to begin to look at the transporters and other maybe fungal specific proteins that might be valuable to examine.
Cool. And then I have another question. This is because you mentioned in your talk that the initial idea to look at glutathione also came from observations made in Listeria. And I was wondering, I mean, I don't think there's much similarity between Listeria and Crypto. I mean, probably both are facultative intracellulars, but other than that, I'm not sure that I can think of something. Is there any relationship between the role of glutathione and virulence of the bacterial pathogen and the fungal pathogen, or is it a completely different setting?
I think the place where the mechanism overlaps is that glutathione influences this PRF transcription factor, which is a regulator of virulence factors in Listeria. So that's kind of the analogy that or the analogous system that there's a transcription factor response to glutathione. And you're right that the other aspect is they're both intracellular in some point in the disease process, and so Listeria is responding to intracellular glutathione from a phagocytic cell or the host cell. Whether Crypto does that in a macrophage, we don't really know, but it would be interesting to check. So, yeah, but the transcription factor binding is probably the key connector where the underlying mechanisms would be similar.
Okay, and just out of curiosity, and perhaps this is going too far, but as you also work on the plant pathogenic fungi, would you assume that also in this setting glutathione play a role?
Absolutely. I didn't go into it, but we've deleted the glutathione, the GSH2 gene from Eustelago as well, and there's a virulence defect for that particular mutant in corn. So there's there's an interesting connection with mitochondria there too that I won't go into, but yeah, we're putting together a story on glutathione in Eustelago in parallel.
Okay, cool. Back to
Thank
you.
you for that. Thank you for that question.
Julia, I have a question about the knock-on consequences of being deprived for phosphate, either genetically or because of the local environment conditions. So one of the things that you mentioned happens is that the phosphate in the wall and those phosphodiester bonds are really important because they are required to build parts of the mannans on the surface. And if you were to have less phosphomannan, there'll be knock-on consequences in a number of ways. So, for example, it's been shown that charge due to the phosphate, the negative charge, affects the sensitivity to inhibitory peptides and proteins, cationic peptides, and also contributes to the overall hydrophobicity of the cell. So those are two additional ways that the fungus might be altered under conditions of phosphate deprivation. And I wondered if you'd considered either of those possibilities that, in terms of the virulence phenotype which emerges under those conditions.
Yeah, that's a great question, Neil. And I want to acknowledge that it was you who suggested looking at phosphomannan in the first place. And that really was a fruitful path to go down. One thing that we were quite puzzled by is how can it be that a phosphate transporter, one of four, has a virulence attenuation phenotype when a host is not exactly a phosphate poor environment. There's lots of different reasons that might be the case that I can touch on, but it could very well be that the cell surface that is the first line, so to speak, of defense against antimicrobial peptides like LL37, cathelicidin, etc., is altered so substantially in cells that are starving for phosphate. And one aspect of the host phosphate connection that I haven't mentioned but that I think is important to think about, is that out of 4 phosphate transporters that Candida has, 3 of them have an optimum in acidic conditions. And that's just because of how the actual transport mechanism proceeds. For example, PHO84 co-transports a proton in order to counteract the electrostatic charge of the phosphate anion. So in the host, humans, we maintain our bloodstream and tissue at pH between 7.35 and 7.45. So out of the 4 phosphate transporters, out of which PHO84 is actually by far the most efficient and capable, only one is efficient at that relatively alkaline pH, which is PHO89. And so in the host, despite the abundance of phosphate, for example, in human cells, it may well be that that the Candida cell is starving for phosphate because its efficient phosphate uptake mechanisms just are not working. And that was explored also in Cryptococcus by Julian Georgievich. So it seems to be a common phenomena for fungi that did not necessarily, most fungi are not ideally adapted to humans, just to say in it. Just end there, because there's a lot more to be said about that.
Yeah, I am not surprised that there's a link with both pH and possibly also calcium signaling, which are because they all come together collectively, don't they? And for example, the accessibility of phosphate, precipitation of phosphate. So that's great. Well, we're getting towards the end. There might be time for Oliver just to squeeze one more question in, but I think probably this may be the last one. And any additional questions, we will make sure they get to the 2 speakers, but will be sent to them, and they might be able to send you a bespoke answer individually. Thank you. Oliver.
Okay, so Jim, you saw it probably in the Q&A. It's a question, but also kind of an application to your lab. And the question is whether you often, if you have an interesting biological phenomenon, study this both in the human and in the plant pathogen, because the student here thinks that this is great, and so do I actually.
Okay, now that's a fantastic question and comment, and it's very insightful, because the way we got into studying Cryptococcus was we had characterized cyclic AMP signaling as a nutrient sensing signaling in Eustelago, and it controls that filamentous, that dimorphic switch from yeast to filaments. So then we went and looked at Cryptococcus in collaboration with Joe Heitman's group and Andy Alspa's group, and there you have the morphological influence of cyclic AMP on capsule. So it's a very good demonstration of the same pathway, same components being used for different kinds of morphological outcomes in two different fungi. They're both basidiomycetes, so it makes it easier to compare, I think. Yeah, great comment. Thank you.
Yeah, okay. So I think with that, we are right at the end. Thank you very much for two wonderful presentations and also a great discussion. I would like to remind everyone that the next MycoTalks is going to be on 31st of October with Gordon Brown and John Taylor. So likely also interesting. And I hope to see you again there. Thank you very much to everyone.
Thank you.